Thanks, Eileen, and thank you to ASM for giving me the opportunity to speak to you today. So we know that anthropogenic climate change and other types of global change have already significantly impacted biodiversity. However, predicting organismal responses to these changes is challenging, in part because responses can vary even among closely related species. Understanding this heterogeneity is a major goal of my dissertation, and today I'll be telling you about the work I did on chipmunks in Yosemite, as well as how my work fits in with a really unique historical data set on small mammals. So my major question of how environmental change will affect biodiversity, I'm going to guess that that's something that many people here are interested in, and I think we'll hear about a diversity of approaches to address this question over the course of this conference. The angle that I took was to select 2 chipmunk species and to study their behavior and ecology. Although my project fit into the span of a PhD, its roots go back much farther, nearly a century, to Joseph Grinnell. Grinnell pioneered concepts such as the ecological niche, and he was also the first director of UC Berkeley's Museum of Vertebrate Zoology. He was very forward-thinking in recognizing the value of the MVZ's collections, particularly as an historical baseline, saying that this value will not, however, be realized until the lapse of many years, possibly a century, and emphasizing that the student of the future will have access to the original record of faunal conditions in California and the West. I'll tell you more about Grinnell's contribution in just a minute, but the important point with this quote is that my PhD work fits into a bigger and longer-term context that can tell us how small mammal communities have changed over the past century. This is an outline of what I'll present today, and I'll begin with information about the Grinnell Resurvey project, briefly, which gives us information on how elevational ranges of small mammals in California have changed over the past century. The key point here is that we know how elevational ranges are changing, but we're less clear on why, and specifically why some species ranges have changed while others have not. And so my contribution towards addressing that why question is to examine how patterns of habitat use can help us understand elevational range changes. So that's what I'll spend most of my time telling you about, and then I'll wrap up with some suggestions for future research in the chipmunk system. So to dive into the Grinnell Resurvey project, this consists of a set of paired historical surveys and modern resurveys of sampling transects throughout California, which are shown in the pink boxes on the map in this slide. The historical surveys were initiated by Grinnell and his colleagues around 1915, and they took meticulous field notes and generated a data set that includes information on the elevational ranges of small mammal species. Those historical field notes were detailed enough that nearly a century later, a group from the MVZ, including Jim Patton, Craig Moritz, and others, was able to resurvey the same sites. So this made it possible to look at how elevational ranges changed over time. These are the results of the Grinnell Resurvey for the Yosemite transect, and each bar represents one species. You can see that there's a lot of heterogeneity in types of responses, with some species expanding their range, those are shown in green, others contracting their range, indicated by red coloring, and others showing no significant range change. This heterogeneity exists even among closely related species. For example, the bars that are circled are all chipmunks in the genus Tamias, so same genus, different species, and they show a diversity of patterns of response. And I was interested in figuring out why such heterogeneity exists among closely related species. To do that, I focused in on 2 chipmunk species because they showed contrasting patterns of response. First, I focused on Tamias alpinus, the alpine chipmunk, which showed an elevational range contraction and is no longer found at lower elevation sites where it was present historically. My second study species was Tamias speciosus, the lodgepole chipmunk, which showed no significant elevational range change in the Yosemite region. Another nice aspect of this study system is that my 2 study species overlap in portions of their range, so areas that appear in kind of the purple darker area at the mid elevations on this slide. And this allowed me to select sites where both species co-occur and to compare what they were doing in areas of overlap. Another strength of the chipmunk study system is that there's a solid foundation of information on their biology, behavior, and habitat associations that I was able to use as context for my work. For example, in addition to information on elevational range shifts, we know that Tamias alpinus is typically found in rocky, exposed habitats, whereas speciosus is often found in more forested areas. But much of this work on patterns of habitat use is qualitative, and it doesn't systematically compare what the two species are doing, how they use habitat, at locations where they co-occur. And so that's what I wanted to add. And just as a side note, this slide also introduces you to the color scheme that I'll use for the rest of this talk, with alpinus in a light blue color and speciosus in red. So my main question was how habitat specialization can shape patterns of range shifts in alpinus and speciosus, and I broke this down into two smaller questions. First, I was interested in how much the two species overlap at a fine scale. So if you go to sites where they both occur, are they using the same portions of the habitat, or are they partitioning microhabitat? And second, whether Tamias alpinus is indeed more of a high elevation habitat specialist than Tamias speciosus. To address these questions, I selected 3 study sites in Yosemite National Park where my 2 study species co-occur, and I also chose sites that had been visited either in the historical or modern Grinnell Resurveys. I visited each of my sites in 2011, 2012, and 2013, and I also made sure to choose areas where there were transitions from more forested habitat, like you can see in the bottom of the slope in the picture in this slide, to more rocky exposed habitat. My methods were to place traps to span those forested to rocky habitat transitions, and that enabled me to capture individuals of both species. I then collared adult individuals and tracked them using radio telemetry until I'd gathered data from 8 to 10 individuals of each species at each of my study sites in each of my study years. These are the results from my telemetry work. And what you see here are polygons. Each polygon represents the areas used by one individual chipmunk, so either an alpinus in the bluish color or a speciosus in red. And there are a couple points that I wanted to make about these results. First, you can see that there is extensive overlap at many of my study sites between my 2 study species. And I think this is important because it suggests that my 2 species are likely to come into contact with one another and that there's the potential for interspecific competitive interactions to occur. The second point is that while there is some overlap, there are also areas at my study sites that are used exclusively or primarily by one species or the other. So the obvious follow-up question there for me was, if you look at those habitats that are used by each species, are they different? And I asked that question at all 3 of my study sites, but for purposes of time, I'll focus on one site today, Cathedral Lake. So my method for assessing habitat differences, I began by downloading remotely sensed Landsat imagery and calculating NDVI, which is a measure of greenness, and in this habitat likely captures a lot of information about tree cover. So the important point is that high NDVI values mean that there's a lot of vegetation, low NDVI values mean that there's less vegetation. So my expectation, based on what I know about the biology of my 2 study species, was that I would see lower NDVI values in areas used by Tamias alpinus, and that I would also see a lower range of NDVI values if Tamias alpinus is more of a specialist. That is, in fact, what I found. These results, again, show data from Cathedral Lake in 2012 and 2013, with alpinus in blue and speciosus in red. And even though I haven't shown you the data from my other study sites, the trends were similar. So what I interpret from this is that Tamias alpinus is found in areas with lower NDVI, so less vegetation cover, and that NDVI varies less for Tamias alpinus, indicating a higher degree of specialization. But a caveat to that is that NDVI doesn't tell the whole story. For example, it doesn't capture some of the differences between tall trees, like the ones in the background of this picture, and shorter, bushier ones, like the ones in the foreground. These differences are certainly relevant for my study species, since Tamias speciosus frequently uses the canopy of tall trees, whereas Tamias alpinus does not. So to go into more detail and describe the habitat, I gathered my own microhabitat data in the field. I focused on areas within 5 meters of a trapping station or telemetry point and recorded information about microhabitat and vegetation characteristics. I input 14 of these variables into a principal components analysis and found that 3 axes were sufficient to explain much of the variation in my data. These show points plotted along 2 of those axes, and I know they're kind of small, but speciosus is in red, and alpinus is in blue. The interpretation of my axes was that the first axis, PC1, captured information about tree and rock cover. So high PC1 scores indicate heavier tree cover, low PC1 scores indicate more exposed rock. PC2 captured information about ground vegetation, and PC3 captured information about downed wood. So once I plotted my data, I then extracted PC1, PC2, and PC3 scores and compared means and standard deviations between my 2 study species to assess how habitat characteristics and the variability in those characteristics differed between my 2 study species. My most important result was that PC1 scores were lower in Tamias alpinus, suggesting that alpinus uses areas with more rock and less tree cover. And that's consistent with what I found in my NDVI comparisons. My NDVI comparisons also showed a lower range of NDVI values for Tamias alpinus, and that's also consistent with my microhabitat data, which if you look at the standard deviations for PC3, those show that Tamias speciosus used a wider range of habitats with respect to downed wood. So to conclude, I'll come back to my main question of how patterns of habitat use can help us understand elevational range changes. And the first part of tackling that question was to look at how much my study species overlapped at a fine scale. I found extensive overlap, suggesting many opportunities for competition, but that overlap was not complete. And in answer to my question about whether there are differences in habitat breadth and characteristics of habitats used by each species, the answer is yes. And in particular, I found that tree cover is important for Tamias alpinus. And that's interesting because we know that tree cover is changing in the Sierras. Over the past century, tree density in many areas has increased, and those types of changes are likely to be relevant for Tamias alpinus in particular. So to conclude, I found first that competition is potentially important, but I really only took the first step towards demonstrating that my 2 study species can come in contact with one another, and I think it would be useful to follow up to conduct more manipulative experiments to really examine the impact that competition has on my 2 study species, for example, by doing something like a removal experiment. Second, I found differences in patterns of microhabitat use, and I suggest that this means that elevational range changes could reflect divergent responses to changing environment. What I mean by this is that Tamias alpinus seems to be more of a high alpine specialist that could be tracking suitable habitat conditions. In contrast, Tamias speciosus seems to be more of a generalist and does not seem to be as strongly affected by the types of changes that have occurred over the past century. In terms of future directions, I've spent my time today telling you about 2 chipmunk species in the Yosemite transect, but the Grinnell Resurvey project includes data on other species, both of chipmunks and other small mammals, as well as data from other regions of California. And this is particularly interesting because we've seen different patterns of temperature and precipitation change in those different sampling areas, and even at different elevations within single sampling transects. And so I think it would add a really important perspective to scale up and examine not only other species, but also other transects in California. Going in the reverse direction, I think it would also be useful to scale down and to really zoom in and look at how changes in the plant community have affected my 2 study species. And I have those analyses in progress, looking at how habitat specialization translates into dietary specialization. And I'm using field-collected chipmunk hair samples to conduct stable isotope analyses of diet, as well as plant samples and information on skull morphology that one of my collaborators has gathered. So to conclude, I wanted to take things back to this quotation that I started with. and say that we've learned a lot about some of the patterns and processes that can explain heterogeneity in range responses, but I still think we have plenty to learn. Enough to keep many students of the future occupied for the lapse of many more years, possibly another century. And with that, I would like to thank many, many people who've given me support during this project in terms of assistance in the field, funding, and thanks again to ASM for giving me the opportunity to speak here. And with that, I can take questions.